welcome to this lightning talk about enabling drug discovery with open targets and open targets genetics. I am Björn Wouters, a data scientist and open targets expert at the Hive. I'm excited to show you all the ins and outs of open targets and how we as the Hive could perhaps enhance your open targets experience. Let's start with a small introduction about our company. We try to enable open science by developing and implementing open source solutions and verifying data in life sciences. Our expertise varies between multiple open source solutions such as CBioPortal for the visualization and analysis for large-scale cancer genomics datasets, Odyssey for harmonizing real-world data, Radarbase for enabling remote research, FairSpace, which is a metadata platform that enables fair by design research, different biomedical data services, but last but not least, open targets, which this lightning talk is about. Let's start with the foundation of open targets, which is the Open Targets Consortium. The Open Targets Consortium is a public-private partnership between academia and the industry that enhances the drug discovery and prioritization process. From the Open Targets Consortium, many data has already been generated, and in order to store and analyze it, together with public data, multiple software solutions are already developed. Two of the main software packages are the Open Targets Platform and Open Targets Genetics. So let's start with the platform. First, there is a public portal available that contains a vast amount of data already. The idea behind this platform is simple. It contains genes, diseases, and anything that associates a gene with a disease or vice versa. The evidence is extracted from over 13 different data sources ranging from mouse models to simple somatic mutations. All this data is stored and accessible within the platform, its web application. Here you can see the disease page in the Open Targets platform. As you can see, the example disease here is Noonan syndrome. In the matrix on the page, you can see all the genes on the y-axis and all the evidence linking these genes to Noonan syndrome on the x-axis. All the data on this page is interactable, so clicking on one of the association will bring you to the associations page. And on this association page, all the different data sources are shown in either tables or visualizations, giving you the chance to inspect them in more detail. Here we look at the evidence page of the PTPN11 gene that is associated with the Noonan syndrome by 8 different data sources. Here we look at one of those data sources in particular, the OT Genetics portal. This data source associates the genes with diseases by GWAS studies. And this brings us to the next application. A whole new application is dedicated to GWAS evidence, mainly because study shows that drugs are over 2 times more likely to succeed if the target is supported by GWAS studies. Just like the Open Targets platform, Open Targets Genetics has a public available portal containing many GWAS studies already. The largest difference is that the Open Targets Genetics associates genes with diseases extracted from GWAS studies, and these variants detected by GWAS are linked to genes via multiple data sources, such as the genomic distance to a gene and chromatin interaction. Just like the Open Targets platform has Open Targets Genetics a web application that makes the data easily accessible and retrievable. Here you can see the study page of Open Targets Genetics, displaying an LDL cholesterol GWAS study. On this page, a Manhattan plot is displayed, showing all the variants that are associated by this study. Next, you might wonder, where does the Hive come in this story? Well, the Hive has a team specialized in everything around open targets, including the code base, the data, and the latest development in the field of both GWAS and just drug discovery. By combining our knowledge with the request of our customers, we can deliver projects like in-house installations of open targets applications, customizations of the applications, for example, adding additional visualizations or changing visualizations that are already there. Also, we could add additional public or custom data. So at this moment, there is a range of public data already there but that doesn't mean that if you have a particular public data structure that you like to have implemented in OpenTargets, that we cannot add this on top of the already existing data. And I can imagine that you have some custom data lying around that you might have forgotten and never looking at, but might even be contains valuable information when adding to additional public data. Next is filtering data. So you could focus on one of your data from your fields of interest. If you, for example, only care about skin disease, we can filter all the genes and particular diseases in your field. Next is adding authentication layer on top of your OpenTargets genetics or platform application. This way you can monitor who is using the applications within your institute, but this means you can also, for example, integrate it with your single sign-on. This isn't everything we can do. We can obviously do more than only this. There are also some application-specific developments. For example, for the OpenTargets platform, you can add any data source of your interest that adds additional information about a gene, a disease, or the relationship between the gene and the disease. For OpenTargets genetics, you can think of a tailor-made GWAS processing pipeline that can process the raw GWAS into valid OpenTargets genetics data, which we can then load and visualize in your application. Next, we also have a side project at this moment. The Hive is working on our own OpenTargets genetics portal, including all kinds of new features we have developed in previous projects. Visit our portal at genetics.thehive.nl to see our custom development. And last, if you have any questions or are interested in your own OpenTargets solutions or other projects from the Hive, don't hesitate to contact us. We are happy to answer all your questions. I would like to thank the Festival of Genomics for having us make this lightning talk, and thank all of you for listening to this presentation. And I wish you all a lovely and healthy day. Thank you.